Okay, our last section is on CRISPR. So if you'll watch this video on CRISPR and then come back and we'll talk about the process. So CRISPR stands for Clustered Regularly Interspaced Short Palindrome Repeats. It is a natural defense mechanism found in bacterial cells, mainly E. coli bacterial cells back in the 1980s. So, if you look at this, this is just something I found from another website kind of explaining what this palindrome is. If you look at it, this one DNA strand is GAATTC. Remember, 5 is top, 3 is bottom. If you look at the other side, it is 5 to 3, top to bottom, GAATTC. So we have the same sequence on both sides. So a restriction enzyme will know to cut at this sequence. So it's kind of like the DNA sequence, like you could see race car is spelled the same forwards and backwards. So what this allows to be done, and it's just a real quick summary here at the bottom with a picture. The first thing we could see here, the target DNA. So this target DNA could be a gene that predisposes someone to have a cancer. It could be essentially any mutation that leads to any type of disease, or something that could lead to a syndrome. And there we see the CRISPR molecule, and this is using a particular type of virus called a bacteriophage, which literally means a bacteria eater. And the second part, if you look at it, once this target DNA has been identified, then the CRISPR enzyme cuts the target DNA at the point found by the guide. Again, the point found by the guide, using enzymes, these restriction enzymes, to cut at these locations. And then 3 is this addition of this healthy or non-mutation, non-cancerous gene into the DNA. So this could be something that could help eliminate or fix issues with the human genome, like those genes, like the BRCA gene, BRCA, that predisposes certain people to have breast cancer, to completely eliminate that. So what you see here are a couple of ladies associated with this CRISPR, this Cas9 system, and some of the challenges that they face, the safety of this. So yeah, there's a target piece of DNA, but what if it goes in and inserts the gene into a non-target piece of DNA? It could happen. Ethical concerns. There's a lot of ethical concerns when it comes to any part of genetic modification. Is we can, but should we? And then, of course, regulatory challenges. So remember that the scientific community has to get everybody on board with something. So just going through this little PowerPoint here, you can see there's a lot of science to get through to through this, to have people see what the benefits would be. Target medications, the example here, tumor suppressors. So those are genes that stop cells from continuing to go through the cell cycle on their own pathway. Biofuels. So these fuels that we could produce, like phototrophic algae, which we would call them probably more autotrophic algae, and disease-resistant crops to prevent from having to use pesticides. And then uh, looking further off into the future, so this CRISPR system going beyond working with playing around with these bacteriophages and bacteria, but what can be done with it. So a couple of pictures that I found here: CRISPR and this Cas9, which is the protein used in a laboratory with this particular enzyme. A lot of uses in agriculture. So, in crops, improving the nutritional value of crops, enhancing disease resistance, farm animals or fish products, improving the meat yield, so making more muscle mass, less fat. Um, and speaking of more muscle fat, so I found this article about muscle gene edit creates buff beagles. And it says the test of the CRISPR gene editing tool in dogs shows mixed results. So, again, kind of seeing what you can do with other animals before we start doing gene therapies and removing genes and replacing them in humans. So, this is done, and there's like a Dr. Lay, and this is what he said at the end of this. It says, Nextly and his colleagues hope to make mutations in beagles that mimic genetic changes implicated in Parkinson's disease, which we know Parkinson's disease of the neurological system in humans, hearing loss in humans, to study those diseases and potential gene therapies for them. But the goal is not just to make a designer pet so that they don't have like bigger, cuter ears. But you can see what they were trying to do here with creating more muscle mass. Those 14-month-old pups, they don't really look like they're completely buff, but some of the things they talked about the article, maybe why we can't see is all their muscle mass is because they have hair. Okay, the pros and cons of CRISPR, and there's a lot of different places where you can go to that are more heavily leaning towards the pros of using this technology to eliminate genes that are unhealthy or undesirable, and the cons against it. So you could look at sites that are pro CRISPR or those that are against CRISPR. So this is just something that I found that shows you a little bit of both. So the pros, modifying T cells to kill cancer cells. So what if we could make it so that our own immune system sees cancer cells as foreign and then attacks cancer cells the way they do the flu virus? That sounds pretty good. Curing genetic diseases. And we saw some things that came along, like genetic diseases from having extra chromosomes. But what about things that come from certain genes anyways, like I was mentioning before, cancer and the BRCA gene, drug development, and improving crops. So we do know, and we saw that with the genetic modified rice, that improving certain crops can make a big difference around the world. And then the cons of this, the germ lining, and the enhancing of people. So I mentioned healthy or desirable. So having, whatever the desired trait would be, maybe being taller, maybe having more muscle, whatever that would be. Bioweapons, so biological warfare, altering something so that, maybe there isn't an antibiotic that can damage the cell membrane and cell wall of bacteria, and they're just undefensible. Reduce genetic diversity. So if we take out some of these bad genes that lead to differences, I mean, just think about it. Mutations led to blue eyes gazillion years ago, but it was a mutation. It didn't kill us, so it was passed on. So if we remove some, then we remove some of our diversity. And then a safety risk. So I mentioned that, yeah, you can have this target DNA, this undesirable gene or this unhealthy gene that you want to replace, but what if it misses and it can't?